In this video, I'm going to be introducing you to signaling cascades and how receptor tyrosine kinases and growth factor signaling hold the door open for cancer cell growth, survival, and metabolic alterations. And of course, a Game of Thrones reference for you. Sorry if it's a little bit painful. Signal transduction is a key mechanism in which cells sense the external environment and then make intercellular changes in order to change their behavior or their phenotypes. In this video, I'm going to focus on two of these pathways. One of them is growth factor signaling through receptor tyrosine kinases, and a related one is survival factor signaling through receptor tyrosine kinases. And these pathways have large changes on cell proliferation, survival, as well as metabolism. Growth factor signaling plays a key role in certain hallmarks of cancer. I know we've talked about the hallmarks of cancer before, but in the case of growth factor signaling, it plays an important role in both sustaining a proliferative signal as well as deregulating cellular energetics and changing metabolism. So the general way that receptor-mediated signal transduction works is that you have a ligand, and in this case that's a growth factor, and when the ligand binds to a receptor that's usually extracellular, you have changes to the intercellular portion of that receptor. And the intercellular portion of that receptor, once it's been modified, then causes a cascade of events to occur in the cytoplasm. And this often leads to amplification, where the receptor affects one protein, but then many proteins are affected downstream. And these are called the cytoplasmic effects. There are also nuclear effects that occur, and changes in gene expression often accompany that. You can also have signal divergence, where multiple pathways can be activated by a single receptor. Because it can be disadvantageous for a cell to maintain this growth factor signaling, we also have negative feedback loops, where a cell can essentially say that we have this pathway active, it's okay to turn it off now. There also can be signaling inhibitors, where a cell can say, well, we're actually not ready to activate this pathway, so we're going to block it at the onset. There are several different types of proteins involved in these cascades, and I'm going to focus on two of the main players, kinases and small GTPases. So kinases are proteins that add phosphate groups to other proteins or to themselves. And they come in two flavors, tyrosine kinases, which take phosphates from ATP and add them to tyrosines, and serine-threonine kinases that phosphorylate serine or threonine. And in most cases, you take an inactive protein, which can be a transcription factor, a kinase, or an enzyme, and when it becomes phosphorylated, it can enter into an active state. In addition to kinases, you also have phosphatases, and these are the opposite proteins that remove phosphates from proteins. So you have a balance between kinases that are adding phosphates and phosphatases which are removing those phosphates. Examples of tyrosine kinases include epidermal growth factor receptor, or EGFR, and HER2, a related protein that's important in breast cancer. Examples of serine-threonine kinases include RAF, MEK, and AKT, proteins that we'll talk about again in a little bit. So when a protein becomes phosphorylated, several different things can happen. One thing that can happen is that you can enable specific phosphobinding proteins to interact with that target protein, which can change the protein's stability, degradation, or localization. If the target protein is an enzyme, then it can become activated or inactivated by the phosphorylation event. Phosphorylation can also change the protein's conformation. For example, it can expose a nuclear localization sequence and cause translocation into the nucleus. The other main players that play a key role in cell signaling are small GTPases. For example, RAS plays a very important role in growth factor signaling. And RAS comes in three forms, HRAS, KRAS, and NRAS. So small GTPases are small proteins that bind to GTP, or guanosine triphosphate, and hydrolyze it to guanosine diphosphate, or GDP. And there are adapter proteins that help push the small GTPases from one state to another, where the GTP-bound form is considered the on state and the GDP-bound form is considered the off state. So for example, GTPase activating proteins, or GAPs, promote the hydrolyzation of GTP to GDP, turning the GTPases into an off state. And this includes P120 and NF1. There are also proteins that help GTPases bind to GTP, These are called guanine exchange factors. An example of that is SOS. So there are many cancer associations with small GTPases. This includes KRAS mutations that activate KRAS in about 30% of lung cancers and 90% of pancreatic cancers. We also observe high levels of guanine exchange factors like SOS in many cancers. And you also observe loss of GTPase activating proteins or gaps like neurofibromatosis 1 in many cancers as well. To learn more about small GTPases, see Tim Formosa's video. So now that you know the main players, let's put these pathways together. So the growth factor signaling pathways start where a growth factor receptor such as EGFR or HER2 is bound by a ligand. And when it's bound by ligand, this causes dimerization of the receptor. And then this leads to cross-phosphorylation of the dimer. Activation of these receptors can also occur if you have overexpression of the receptor proteins or if you have activating mutations. The phosphorylation of the receptors then causes activation of SOS, which is a guanine exchange factor. This in turn activates RAS and promotes the GTP-bound form of RAS. The GTP-bound form of RAS can then bind to RAF, and this activates RAF, which is a serine threonine kinase. And then this activation leads to a number of downstream events, all involving kinases, that leads to active MAP kinase signaling. So we call this pathway the RAS-MAP kinase pathway, and this promotes proliferation of the cell. The activation of the receptors also leads to activation of the PI3 kinase pathway. This leads to activation of PI3 kinase, which is a lipid kinase. And this lipid kinase, when it performs activity, also activates AKT, which is another kinase. And this leads to the activation of the PI3 kinase AKT pathway, which leads to survival and metabolic alterations. There's negative feedback on both of these pathways. So for example, neurofibromatosis 1 is able to block RAS activity on the RAS-MAP kinase pathway. And in the PI3 kinase AKT pathway, we have PTEN, which is a phosphatase, which opposes the activity of PI3 kinase. So if we look at the kinases in this pathway, we see that there's several kinases that are important. That includes the growth factor receptor itself, as well as RAF and MEK on the RAS-MAP kinase side, and then PI3 kinase and AKT on the PI3 kinase side. We also have a phosphatase, PTEN, which plays an important role. The main GTPase that plays a role in growth factor signaling is RAS. And it's also affected by its adapter proteins, SOS, which promotes its activity, and NF1, which blocks its activity. And these pathways are very important in a number of different cancers. As an example, in the first TCGA paper on glioblastoma, they found mutations in many of the different pathway components. And the activating mutations are shown in red, and the inactivating mutations are shown in blue. So this is to highlight the importance of this pathway in cancer.